Okay, so thank you very much for your kind introductions. So I'm going to talk about biological functions and functions of modules originated in structure of the reaction network. So let me first introduce my personal interest. So I have been interested in dynamical behavior of complex network systems in biology. So of course, we know that there are many examples of the network systems in biology, including so large number of the biomolecules and interaction between them. And based on such complex network, dynamics of the molecular activity runs, and dynamics are origin of the biological functions, we believe. So we want to understand dynamics based on these complex network systems. But of course, it's not easy to understand the dynamics of such high-dimensional complex systems. So we usually have to develop mathematical models under the assumption of functions or parameter values. But such a modeling approach is not so efficient if system is so large. So we have developed our original method. So we developed theories which derive important aspect of the dynamic behavior from information of the network alone. In other words, model free. So we do not have to determine all of the dynamic behavior in detail. So we can determine some aspect just from network. For example, we can determine key molecules from regulatory network, and we can determine or mechanism of the regulations by enzyme activities from reaction network, and we can even determine bifurcation properties from a network structure. And today I'm going to give you a story based on the this second theory.
There are a
So
lot of
let me can I? Okay.
yeah.
So let me
So
first introduce biology of uh, reaction network. So this is one example, famous example of reaction network. So this is carbon metabolism systems from E. coli. So it is very general under universal systems, and the basic structure is shared between a human being and bacteria. So organisms obtain a chemical energy in the process of the decomposing sugar in these systems. And in this system, each arrow indicates a state transition induced by reactions. For example, here, sorry, I should change. So here, alpha glucose is transformed into beta glucose via reactions, which is mediated by GAL-M enzyme. Okay, and beta glucose is a product of this reaction, but at the same time, it is substrate of another reaction. And in cells, multiple reactions are connected by sharing product and substrate, and connected to form network. So this is the meaning of the cell reaction network in biology. Okay. And how organisms control the dynamic behavior of a reaction network? It is basically by changing activity or amount of the enzyme. For example, enzyme can change expression level of the enzyme gene, and change in amount of the enzyme may influence concentration of the substrate or product chemical. So this sounds reasonable, but so this is just an intuitive argument. And I will explain how this process is interesting and difficult to understand. So before explaining my theory, so let me briefly review how dynamics of the chemical reaction has been studied. So of course, there are so many theoretical studies, but basic idea is very simple. So change in concentration of each chemical in a system is that determined by difference between influxes and outfluxes. For example, in these small systems, chemical A increases with fluxes of reaction 1 and inverse 2, and decreases with the flux of reaction 2, and which is shown in this ODE. And these relations between change in chemicals concentrations, and the differences between fluxes can be summarized by introducing a matrix which is called stoichiometric matrix. Okay, and I called flux, but flux is also called reaction rate, and the reaction rate function is often assumed by using often modeled by assuming so Hill functions or Michaelis Menten functions or mass action kinetics. But in my talk, we do not assume any detailed mathematical formula for reaction functions. Okay, we rather use two trivial facts. The first fact is dependence on chemical concentration. For example, reaction 2 connect from A to B. So reaction rate of the reaction 2 should depend on the concentration of the chemical A, UA. Okay. So in addition to substrate chemical, some chemical may regulate reaction rate of the reaction 2. In that case, we integrate such regulator chemicals into the argument set of the reaction 2. So dependence on substrate or regulator is the first assumption we use. Okay, and the second assumption is reaction parameter. So each reaction function has a single parameter which corresponds to amount or activity of enzyme. For example, R1 possesses K1, and R2 has K2, and R inverse 2 has K inverse 2. And now we consider a problem. How change in reaction parameter K influence steady state concentration of each chemical, UM. Okay. So now I explain our theory, structural sensitivity analysis. So let me first claim our message. So we can determine qualitative response of reaction system. I mean, which one increase, which one decrease, or no change, can be determined from structure of the network alone. Okay. So we start from steady state of the previous dynamics, steady state of these equations. And of course, I'm sorry, steady state, both before and after perturbation. Okay. So of course, change in reaction rate vector delta R is in the kernel of the stoichiometric matrix. Okay. And from now, we call basis of the null space of stoichiometric matrix cycle. So following the data, tradition in this field. Okay. And we take total derivative of this equation with respect to parameter change like this. Okay. And just by taking linear algebra, we obtain matrix formula like this. And it looks complex, but not so difficult. So this is just summarize into unknown part, this part, and known part. And the right-hand side is diagonal. So our method is just two steps. First, we construct a matrix from structure of the network. Then we calculate inverse of the A matrix. And in the inverse matrix, each entry implies response of each chemical A, B, C, D to the perturbation of this reaction 1, 2, 3, 4, 5, 6. Okay, and this is yes, I will explain. It's an important point. So this is example of A matrix corresponding to this example network, and you can see there are many zero entries. So actually, a pattern of the non-zero entry in A matrix directly reflect structure of the network. Okay, and so I explained. So each entry in inverse matrix is response of the each chemical to the perturbation of each reactions, and you see. So zero or non-zero of each entry in S matrix is determined from pattern of the non-zero entry in A matrix. And so in many cases, we can even determine plus or minus of each entry in inverse matrix just from the pattern of the non-zero entry in A matrix. So this is meaning that we can determine a qualitative response from structure of the network. Okay, I will explain. Simplest example. Oh, I'm sorry. So this is just actually simpler version. So in some cases, a system may have conserved quantity, which are identified by left null space of stoichiometric matrix. In this case, we need another set of equations like this corresponding to conserved quantity. So by combining previous set of the equations and also this set, we have little bit larger matrix formula, but the essence is the same. So by after constructing a matrix and also calculate inverse, we obtain a response of the system to the perturbation. Okay. So this is simplest example. So we have an input flux with chemical M, A, and after two steps reactions, chemical C goes out from the system. And let us consider to increase enzyme activity of reaction 2 corresponding to the first step. Okay, so you have gene of the enzyme, so mediating reaction 2, and you increase expression level of this enzyme. And what will happen as a steady state? Okay, I think most of the people consider that. So then flux of the reaction 2 should increase, and the downward chemical may increase, B or maybe C. Actually, such intuition is completely incorrect. So this is our analysis, and our analysis implies that. So only the substrate chemical show the change at this state. So it should decrease in its concentration. And there should be no change in downward chemicals and no change in fluxes. Okay? And our theory is model free. But by assuming mathematical formula for reaction rate, so we should observe this kind of the response always. So actually, after increasing enzyme activity or amount, we should observe increase in flux of reaction 2 and the downward chemical, but they are just temporal. So they soon go back to the original level. At the same time, we should observe decrease in substrate chemical A like this. So we can understand that. So increase in enzyme activity is compensated by decrease in substrate chemical, and there is no change in downward. Okay. So important point is that so we do not have to calculate dynamics. Just by calculating inverse of this small matrix, we can determine the final states. Okay. And let me briefly explain another, a little bit non-trivial cases. So this may be actually very difficult. So now, so we have feedback from chemical C to chemical A. And chemical C has two output arrows, 4 and 6. And let us consider two perturbations to each of these two output reactions, 4 and 6. Okay? And when we increase activity of the enzyme corresponding with reaction 4, in this case, we will observe direct increase in flux of the reaction 4 and that of 5 to 3, and increase in chemicals on the pathway DAB, but C does not change. Okay, and when we increase activity of the enzyme of reaction 6, in this case, 6 is silent, and we rather observe decrease in flux of reaction 4 and that of 5 to 3, and decrease in chemicals on the pathway D, A, B, and C. Okay, and someone may consider that. So between left and right, similar part changed, but please be careful. In left, C does not change, and in right, chemical C does change. So we gave locally symmetric perturbations, but we obtained very different responses between left and right. Okay. Now, actually, so these yes patterns in response can be understood completely by our finding, which we call law of localization. So, yes, chemical reaction may be very large, but let us focus on substructure. And if substructure satisfies these topological conditions, it has special property in the sense in the behavior of the sensitivity. So let me explain one by one. So focusing on substructure implies taking a subset of the chemicals and subset reactions. And the first condition implies that. So after taking a chemical subset, all of the emanating allo from the selected chemicals should be selected into it, reaction subset. Okay. And the second condition is interesting. So let us count the number of the element in the substructure, the number of the chemicals, number of the reactions, and the number of the cycles, and the number of the conserved quantity. So if these four integers satisfies these equations, then the substructure is buffering structure. And what is buffering structure? So any perturbation inside a buffering structure influences only inside chemical concentrations and inside reaction fluxes. And the outside should be silent at steady state. And someone may realize that so this index is similar to Euler characteristics. So actually, so Euler found that so this index govern homology of the polyhedron in 18th century, but we found that a similar index governs extent of the parameter influence in chemical reaction network. So this may sound something mysterious, but actually proof is very simple. So buffering structure actually corresponds to a square non-zero block in A matrix. And this part should be zero or from output complete condition. Okay. And so you know that. So if square matrix has this structure, then inverse should have the same structure. Okay. And the zero block here in inverse matrix implies responses outside to the perturbation inside of buffering structure are always zero. So actually the proof is very simple. And we could even prove the inverse theorem. So law of localizations claims if local substructure satisfies topological conditions, then we should observe confined response in the sensitivity of the parameter. And so inverse theorem claims that if we observe confined response in parameter sensitivity, then there should exist corresponding buffering structure. So we can connect topology and behavior directly. And let us go back to our example. So in example one, this is buffering structure, one chemical, one reactions, no cycles or no conserved quantity satisfies our conditions. And in second example, this is buffering structure, three chemicals, four reactions, and one cycle, no conserved quantity. And the right case, this is buffering structure, four chemicals, and five reactions, and one cycle satisfies our condition. Okay. An important point is that so local substructure can obtain so special property of the regulatory module just by satisfying these so topological conditions. So I think so this should be very important. So in the biological mechanism or so in evolutionary biology. And now we are looking for examples where buffering structure take important role in actual biological functions. And we think cell cycle is one of such examples. I think you know cell cycle very well. So in cells, there are in life of the cells, there are multiple phases between one cell divisions to next cell divisions. In case of yeast, there are four phases: G1 phase, S phase, G2, and M phases. And the transition between different phases are strictly controlled, especially in 2 points between G1 and S and G2 and M. So cells can invade S phase only when a special complex called cyclin CDK complex, so which we call G1S complex increases. And in another transitions between G2 and M, so cells can go to M phase only when another protein complex, G2M complex, increased. Okay, and these two complexes, G1S and G2M, are different protein complexes, but also they share a common protein, CDK. And it is well known that amount of the CDK is kept constant, well kept constant in the process of the cell cycles. So it is very interesting systems. So this is network for protein synthesis and decompositions and complex formation. And this is G1S complex, and this is G2M complex. And there are 6 species of the protein complexes which include CDK. So these red marked protein complex amount of these red marked protein complexes should be kept constant. Okay, so there are some argument which claim. So if G1S complex increases, then another regulator complex, G2M, may decrease. Okay, I think so. It's interesting. So let us confirm whether this kind of behavior will appear by using structural sensitivity analysis. So we usually read sensitivity matrix vertically. So which parameter influences which chemicals. But let us see horizontally this time. So this is G1S complex, and this is G2M complex. And so examine which parameter influences amount of each of these regulator complexes. So let me show you on the graph. So there are two reaction parameters which influence amount of the G1S regulator complex, and there are 6 reaction parameters which change amount of the G2M complex. Okay, and they are completely disjoint. So disjoint implies that this argument, competition between G1S and G2M, is not correct. So we rather claim so all these two complexes should be independently regulated. So I will show you demonstrations by numerical simulations. So we calculate dynamics of these systems, and while we calculate, or we change a parameter of the synthesis rate of SIG2, so which is component of this protein complex. And we observe, by changing this synthesis rate, we observe change in G1S complex like this and like this. Sorry, so I'm plotting steady state only, okay? But G2M is silent at this period. Then we change a synthesis rate of another cyclin, C13, which is component of G2M complex. And we observe change in amount of the G2M like this. But G1S is completely silent. So this is the meaning of the independent regulation between two regulator protein complexes. Okay, and this independent regulation is actually realized by buffering structures. So this is the largest buffering structure, including G1S complex, which does not include G2M. So even if there are parameters which can influence G1S proteins, they never influence G2M. And similarly, this is the largest buffering structure, including a G2M complex. So even if there are parameters, they can influence G2M, they never influence G1S. Okay, so a little bit complicated. So let me show you, summarize using the simplified cell cycle systems. So there are two important buffering structures in these systems. So blue buffering structure implies that So parameters, for example, synthesis rate of CDC13 corresponding to reaction to influences G2M complex, but it never influences G1S. And also, there's another important buffering structure, green buffering structure. So in this, yes, reaction parameter, for example, synthesis rate of the SIG2 reaction 1 influences G1S complex, but never influence G2M. So I think it's worth to verify whether so these beautiful regulations actually exist in real cell cycle systems. Because so this result is based on the database network, and we do not know whether real network systems has the same yes network. Okay, the database network may be incorrect. So this is verification experiment. So it is collaboration with experimental biologist Professor Aoki in Kyoto University. So prediction says that by increasing CDC13, we should observe increase in G2M, but no change in G1S. And this is the experimental result. So you can see increase in G2M, but G1S is, yes, clearly no change. So very beautiful result. So we can confirm that the blue buffering structure does exist. Okay. And let us see another buffering structure, green buffering structure. So prediction says that so by changing the SIG2 synthesis rate, we should observe a change in G1S, but there should be no change in G2M. And this is the experimental result. So we observe both change in G1S and G2M. Okay. So it is contrary to the predictions. Maybe a little bit unfortunate, but please recall that structural sensitivity analysis is model-free. So it implies that if the network information is correct, we should observe predicted behavior. But actually, we didn't observe. So this implies that the database network is incorrect. So we have to revise the network. Okay. So yes, and we examine yes all of the possible modification of the network. And what we should do is that the blue buffering structure should exist, and the green buffering structure should not exist. Okay, and there's only one possible yes modifications. So only the addition of the CDC13 degradations reactions destroy the green buffering structure. So this one. Okay. And it is also case. So if we consider original complex network. Okay, and please note that this red arrow should be from this monomer CDC13, not from this, nor this from this. And we examine these predictions. Yes, we have to be careful to setting experimental conditions to observe the degradation rate of the CDC13 monomer. And I do not explain in detail. So please focus on this figure. And the blue line, blue curve, and also blue dotted curve corresponding to the change in amount of the CDC13. And this result clearly shows that the degradation rate of the CDC13 monomer is significantly high. Okay, but this is new finding. So previously, it was not previously known. So we can predict unknown reaction from the comparison between theory and experimental observations, and so we can confirm predicted existence of the predicted reactions, and so we could update the network information. So this is summary of the talk. So we can determine response of the chemical reaction network systems to the perturbation in enzyme parameters and from network structure alone. And the buffering structure is very important to specify the regulatory module. And so yes, our theory is model-free, and we can derive important information by comparing theory and also experiment. And in the case of the cell cycle systems, so we obtained multiple results. Okay, this is the end of my talk, and thank you very much for your attention. I'm very happy to receive your questions. Thank you very much.